Well, thank you all for sticking with it here at the end of day 2. But before I begin, I want to give a shout out to John, our very lovely MC, not only for your humor, but your kindness and compassion. John, let me give you an applause. We've all been to many of these seminars before, but I've never been entertained and engaged as you. So keep it up, whatever it is you're doing. I love it. So friends and colleagues, I've met a lot of you here as we wrap up our session together. And thanks to Ruben and all of you in the foundation for bringing us to Phoenix together. I want to pick up where my friend and colleague, Dr. John Mascarenhas, started, which is to finish the story on the new targets, the evolving targets, with specific focus on the combinations that are out there. And of course, as you know, we're all clinical trialists and we work with all the companies out there. My goal here is to bring you the cutting edge stuff, many of which is not published yet. So as you've heard from my colleagues in these 2 days, man, let's take a minute to just hear and listen to, what we've been trying to absorb together. This list is just some of the things that are out there. In polycythemia vera, we heard about the ropeg interferon, which is known as Bezremi, the interferon field really pioneered by Dr. Silver and Dr. Kalajian, both of whom have been here in person during the meeting, the PTG 300, the rusfertide, which John just mentioned. Even now in ET, we've got some movement. In our ET breakout session, one of you nicely asked about the Bometamstat, the LSD1 inhibitor, an oral drug that's in clinical trials. In the even ultra-rare myeloproliferative neoplasm, systemic mastocytosis, some of you may have, we didn't mention it much here in this meeting, but there's a lot of new drug development going on around the KIT inhibition. And then even the more ultra rare malignancies, for those of you out there, there's something called the 8p11 or FGFR myeloid lymphoid neoplasm, pemigatinib. This is a drug that my friend and colleague, Dr. Serge Verstovsek, just pioneered to FDA approval. And it may only affect a dozen to 100 people a year, but it shows us two things: that we're missing the diagnosis of rare diseases, so we're not looking for them. If we don't look for them, how can you diagnose someone with it? And also, if you focus your time and attention on single discrete targets, oftentimes you can develop rational therapy, and then you can expand that into other areas. For example, this drug, can you believe it, is also used in the treatment of gallbladder cancer, cholangiocarcinoma. And then, of course, we'll focus our comments together on myelofibrosis here. Some of these new targets you're hearing about in the JAK inhibitor space, to remind you, ruxolitinib is now 10, 11 years old. Then you have the fedratinib, which we've heard about August 2019. And then pacritinib, the newest JAK inhibitor on the block, exactly this month, 1-year anniversary. A really amazing improvement to our armamentarium. As you know, we can give that drug in the setting of low platelets. And that drug was pioneered by Dr. Mascarenhas, who's here with us today. And then the momelotinib agent, which aims to be the 4th JAK inhibitor. Not yet FDA approved, but under review. We hope to find out in the summer. And we heard about that from Dr. V yesterday. I think the novel combinations, which I'm excited to present to you in my remarks here, I'm going to focus them on what's practical for you right now. There are 3 separate combinations in myelofibrosis that are in what's called the phase 3 testing, as we heard, the late stage of development. And I'm proud to tell you that all 3 of these are global involvement, so international, despite pandemic time, have continued to stay open and enroll, and are available at more than just the usual 1 or 2 academic sites. So they're trying to extend to a place near you. And then finally, this concept of looking at beyond JAK inhibitor altogether. As John showed us, there are some new targets out there. And then the anemia of myelofibrosis. Many of you suffer from. What are we doing about that? So let's get into a team together. The first concept here is that of disease modification. Dr. Mascarenhas brought this to our mind earlier this morning. I think this is something that's the holy grail. I don't think we know how to define it together, but we know it when we see it. I think that the definition of this is evolving, but as John said, let's think of disease modification as something that matters to you, the patient, so overall survival, quality of life, not progressing to a leukemic state, something that matters for the clinical trial parameters so that we can use it from patient to patient, spleen size, symptom score, something reproducible, and then something that means something in the laboratory scientific venue, which shows that the drug is doing what you set out to do: clinical correlatives, cytokine, mutations, all of these things we heard about. So we put this position paper out there last year, and I think it's been well received, to just try to say, if we're aiming for disease modification, why not go for it at the start of your diagnosis? We heard from Dr. Gowan that there are things outside of the pharmacological venue that we can do, lifestyle modification, concentrating on the entire experience of wellness from that time to before we get to the advanced state, so disease modification. With that in mind, the JAK inhibitor monotherapy for those of you with myelofibrosis has simply revolutionized our field. We know that. That's why we're meeting here together. But we have not cured, unfortunately, many of you with this single pill approach, either once or twice a day. And so if we try to go for a longer lasting remission, a cure, something to eradicate the disease from the mother cells, the stem cells they start, my argument to you in our time together is that we probably need to move to combinations of therapy. But before we get excited to do that, we must show you that we're not adding too much to your toxicity burden, that your quality of life doesn't suffer while trying to get to a good goal, and that there are not new or weird or unexpected cancers or side effects or something that pops up while we're doing that. We are starting to see the glimmer of hope from that, and this slide gives you some sampling of that. In the early days, we pioneered the combination with the hypomethylator agent, that's azacitidine. I think we mentioned that here. That's usually used in a cousin disease called MDS or an AML, and it's been around for about 20 years, and combining that with the JAK inhibitor has shown activity in those of you who have a little bit more advanced disease. So that's out there. But these new agents, and I'll focus on the three highlighted, BCL-XL aims to, that's another pathway, that's the agent called Navitoclax, PI3 kinase, and then of course the bromodomain or BET inhibitor. And I think all these other combinations that we have here are in the earlier stages, phase 1 and 2, and I'd like you to keep your mind on that. Man, as we get past the JAK inhibitor space, this can be its own separate 2-day seminar now. We and others around the world have tried so many targets, some of them borrowed, some of them from other diseases that you've heard of, maybe the more common diseases, some of them specific to MPN and myelofibrosis, and some in this new area called microenvironment or tumor hypoxia. What the heck is that? Well, if you think about the bone marrow, the stem cell niche, where these cells are growing, these cancer cells, there's nutrients that are needed to go to the cancer cell unexpectedly. There's blood vessel supply. There's other factors that are there to grow the cancer and to dominate and take over your healthy cells. So if we can attack not only at the headquarters, but at the field sites, the surrounding network of these cancer cells, can we then put these myelofibrosis and bone marrow failure states into remission? And so you see the list, and I'm proud to tell you it's growing, and many of my friends and colleagues here are leading those efforts with all of you, with our colleagues in pharmaceutical industry, and with patients and advocates such as yourselves. So let's get into the details. I think with that overview, I wanted to focus on these three phase 3 trials, so bromodomain, PI3 kinase, and BCL-XL. The first that's really generating a lot of excitement in our field that I'd like to bring to you is that of bromodomain or BET inhibition. So all of these are new kind of pathways that are being revealed that are governing bad cells to grow over good cells. So let's look at this BET, bromodomain extraterminal domain protein. This drug is called pelabresib. It used to have the letters and numbers CPI 0610, and it's already gone through the phase 1 and 2 trials, which is as a single agent in the relapsed refractory setting, and now is in the frontline randomized phase 3 clinical trial, this drug plus ruxolitinib versus only the ruxolitinib. This is interesting. If you picture these myelofibrosis cancer cells, they need a lot of ammo. They need a lot of juice, a lot of firepower to be able to take over the bone marrow. And this is showing you some of that here. We talked about inflammation with Dr. Gowan. You'll hear about it more from Dr. Fleischman in a moment. This tissue injury, this cell injury that gives rise to the disruption in the bone marrow. So now you've got bad cells arising, and they have to co-opt or hijack all these other pathways to be able to be immortal. So these cancer cells, your myelofibrosis cells, are growing and dividing when healthy cells are dying appropriately. This aberrant erythroid differentiation reminds us that these cancer cells can cause red blood cells, the parent cells to platelets known as megakaryocytes, and other cells to grow too much, and then ultimately to either scar over, causing the fibrosis, or to be so hyperactive that this cytokine environment is causing a war inside your body. That's what we talked about in our breakout session. I hate it when people tell you that you don't, quote, look like a cancer patient. What do they know? What do they know about what's going on in your body? That's why it's called internal medicine, right, guys? So it's not external medicine, right? So this sample right here is a new pathway beyond JAK inhibitor that tells you that there's something else going on inside of your body, maybe why you're not feeling well, maybe why you have too many cells or too few healthy cells. So, and I'm really happy that John just talked to us earlier today. We really have the leaders in our field together in this auditorium. So what John did is led this trial so far that shows that the combination of this pelabresib, this new agent I told you about, combined with the Rux, the old agent, is now not only showing safety, it's a well-tolerated oral drug, but at our traditional 6-month time point is actually showing that the responses in many of the patients are staying. It's durable, and it's safe even beyond that. And this is remarkable for a new drug. Just to quickly show you, this is 84 patients that were treated at the time of this data cutoff, and this study is called Manifest if you want to look it up later. This is one of the arms, the combination arm. And what they showed is that it's a real-world distribution of patients. Mean age is 67, fine. We know that's around our average age for myelofibrosis. And the subtypes and the blood counts reflect what a lot of you may have when you were first diagnosed. But wow, look at the bottom. This HMR, high-risk molecular mutations. Guys, now that we're checking this on everyone, a third to a fourth of our patients will have these. ASXL1 mutation you heard about, for example, may portend that you have a higher chance to go to leukemia, maybe even a worse outcome overall. So do these drugs work in that setting, and if you have triple negative disease? So out of these 84 patients, this is a nice diagram that you can sort of quickly look over. It just shows you what happened to the patients over time. As you would expect, patients discontinued for a variety of reasons. In this study, only two patients went to transplant, one patient died. Some patients will come off for withdrawal, toxicity, et cetera. So as I think, John, you were mentioning, it's always good to look at this if you ever get a chance to see clinical trials. What happened to the patient? So overall, this is what we would consider standard for our field. But boy, this is what I wanted to show you. This is this vaunted waterfall plot. How did each individual patient do in aggregate? And amazingly, this combination, when offered up front, is showing that 68% of the patients had what we call the SVR35 at week 24, so the kind of primary endpoint. And if you compare that to the COMFORT studies led by Serge Verstovsek and Claire Harrison 11 years ago, that was in the 40% range. So this is actually a 20 to 30% increase on that. So we have to justify, why am I asking you to take a second pill? It's going to have more toxicity. Well, you're seeing that this could be one reason, that you have a higher rate of spleen. What about the symptoms? Well, when you look at the symptoms, you're looking at, I think, 56% on this study, and that's higher, again, as well than the COMFORT studies. So the combination, at least this top-line data I'm showing you, already shows that it's fairly safe. Most of the patients are able to take it. That's important, and that you're getting much higher than we would have expected with just JAK inhibitor alone. What's happening at the level of the microscope, the bone marrow fibrosis? This has been another holy grail for all of us in the field. That's starting to reverse and reduce in many of these patients, and early on, not over years and years and decades, but within these 6 months, first 1 year. And so what you're starting to see, team, is the start of disease modification, combination therapy to try to really overcome and improve from ruxolitinib or JAK inhibitor monotherapy. Every new agent, we must together look at the toxicity and side effects. The excitement has to be coupled with how does the patient actually tolerate this drug in their house and in the clinic. Overall, as you let your eyes glance over this is a pretty safe and well-tolerated drug. Like a lot of these new agents, it does have some GI side effects, which, working with each of the patients on the studies, we have found to be very manageable. And obviously, some patients, the blood counts can go down a bit too far, so that's called thrombocytopenia, low platelets, again, very manageable with dose modifications and monitoring on the study. So let's just put that in our hat. That was the first of 3 that I'm going to show you. So JAK inhibitor combined with bromodomain or BET inhibitor, pelabresib. That's an extremely important program that's moving forward now in randomized phase 3 registrational study. So this is category number 2, our second bullet point. This is BCL-XL, another one of these pathways inside these cancer cells. This is an important one because I'm involved in it, okay? That's why it's important. John likes to use the word bias, but this is very important. So take pictures and notes here. So obviously, this one is interesting for 2 reasons. So the bromodomain that I showed you that John is leading and this Navitoclax that I'm helping to lead, neither of these are approved yet by the FDA. These strategies have been around for a while, but they have not yet been approved. So that tells you that they're still trying to find the right home, right, guys? So they've been tested in leukemias and lymphomas, right? But the key seminal discovery was about a decade ago that this pathway may be the one that's more upregulated or out of control as compared to the BCL2 pathway, which is the one that Navitoclax, if you've heard of that drug, is FDA approved for. So can you believe it? Two pathways, basically the same name, slightly different. One is a blockbuster drug in leukemia. The other one yet has not been FDA approved. In myelofibrosis, for some reason, it appears that this BCL-XL is the one that's upregulated. And in the lab, first, it was shown that if you use this Navitoclax oral agent, either by itself or in combination with JAK, that you can make this pathway calm down, block it, and then the cells can grow and divide peacefully, happily, as they should, right? Normal cells should grow, divide, and die. Cancer cells are immortal. So we and others, with Claire Harrison, our colleague from the UK, we set out to do a phase 2 trial in giving this to patients in combination approach. But specifically, we did it in patients who already were on a JAK inhibitor, and then we asked you to take the second drug. That's called an add-on approach, and that's a new part of this field. Well, these results were positive. We were able to publish it in one of our major journals, JCO, which just came out, I think, last year. And what we showed among these 32 patients is something very important that I'd like to share with you. The first thing is that the overall survival at the 2-year point of these very patients who've already shown that they're not responding to the JAK inhibitor, but maybe deriving some benefit, we kept them on and gave the second drug. The overall survival, I'm showing you, this is called a Kaplan-Meier curve, and each hash is an event there. What it shows you is that the overall survival was not reached, the median overall survival. So these patients, unfortunately, the historical expectation is some of these patients could have had an overall survival of less than a year and a half, 2 years. Small group of patients, very specific group I just told you about, but this drug, people were able to take it for a long time and able to improve their overall survival. So disease modification? Question mark, maybe. Well, what's this drug doing? What it's doing is it's trying to stabilize this cell death pathway. John nicely mentioned it, apoptosis. Cells are supposed to die. That's their fate. Programmed cell death, apoptosis. But in a cancer cell, they don't. And so this drug aims to restore that homeostatic balance. Interestingly, when it's combined with ruxolitinib, the JAK inhibitor, in the lab and in patients, it goes very well together. The only thing we have to watch out for is that, again, the same signal that you heard before, some GI side effects, and then it can lower the platelets. So again, when you introduce a second drug, you got to watch carefully those blood counts. The other question I had with this drug is, okay, if I care about this disease modification, how do we actually show it beyond the spleen, the symptoms, and the overall survival? So we looked at it with our scientists. And what we found from this refined phase 2 that I just showed you, we published the clinical results of, is that surprisingly, amazingly, at the biomarker level, all of the markers that we wanted to hit are being reduced, reversed, and being hit appropriately. And those markers are interesting. They're called cytokines, these protein messengers we've been hearing all about, some of the molecular mutations, and then ultimately some of the bone marrow fibrosis. Not in every patient, of course, but in a lot of them. And so what we said is if you're actually able to have a combination that can improve your overall survival, decrease your spleen, improve your symptoms, not be that toxic, and at the biochemical level show that you're reversing marrow fibrosis, allele burden fraction, I think we have the start of disease modification. That's what I would like to present to you here. And so what we've done with this trial now is moved it also into the frontline setting. So again, patients, instead of getting the JAK inhibitor alone as you would in the standard, let's give you the combination. Again, another 32 patients, so a different 32 patients. And you can see the waterfall plot. Almost every patient had major benefit from the combination. Again, more than you would expect with the JAK inhibitor alone. And again, it was found to be safe and manageable with the thrombocytopenia and the GI side effects still being the most important in the frontline setting. These responses, team, are regardless of how high risk the molecular mutations are. What about, did patients have bone marrow fibrosis in the frontline setting reduction? Yes, they did. Several of them did, not all, but about a fourth of them had it, and they had it quickly. And what about this variant allele fraction that we've been talking about the last 2 days? Several patients had a reduction of greater than 50%. Again, these are early data. This is a small group of patients. And so, as I told you earlier, this has now moved into the phase 3. The Transform 1 and 2 studies are big global randomized phase 3 studies. Transform 1 is this approach, frontline patients, so Rux Navitoclax versus Rux alone. And then the Transform 2 is in the relapse setting. And we eagerly await those data. Maybe we'll even have those readouts by this time next year. And then a third category. So first category, bromodomain combination. Second category, BCL-XL combination. This is the third and final of the big three. This is called PI3 kinase inhibition. You may have heard of this one. This one's been in solid tumors, so breast cancer, lymphomas, some press in the news. Some of these different agents have had some issues with infections and toxicities, some haven't. But this particular PI3 kinase has been moving nicely into myelofibrosis, and that's called parsaclisib. And so this combination was shown by our friend and colleague, Dr. Yacoub, at the ASH meeting as well. So all three of these, I'm showing you the latest data from December. And again, this should be the add-on approach. So patients already on the JAK inhibitor, and now they're getting the new drug, parsaclisib. And again, as I've shown you with the first two themes, again, you have to see that you're getting some spleen volume response. This one they interestingly did early on, so at 12 weeks. That's at 3 months. And you have to also make sure that this drug is safe and the symptoms are improving. And again, you see that here, that almost all the patients will have some symptom score improvement. We like to say TSS 50, which is actually the 50% reduction. And so what about side effects with this new drug and blood count levels? Overall, what we expected in the early settings, that the platelet levels and hemoglobin are okay when you add in the second drug. So no new or unexpected toxicities that we weren't primed for. What are the toxicities of this parsaclisib? You can see here, a lot of these drugs, it did come out in our breakout session, sometimes either the JAK inhibitor alone or the new drug, you have to look out for immunosuppression, infections, pneumonia. That's something that's part of our field, and you'll see that in all these studies. We live in the time of COVID-19, so you want to look and make sure about that. But overall, this is, again, what you would expect in one of our studies for the expectations of the toxicities. And so I wanted to leave you with this provocative idea. We've been talking about this word for 2 days, this disease modification. I've made to you some arguments here today in this forward-looking talk. You've processed and absorbed the standard of care, and now Dr. Mascarenas and I are showing you some of these new things that are not in the lab, right? These are active things in clinical trials at the advanced stages. We're starting to see more and more groups write about this disease modification. I showed you our position paper. The second one is from a European group talking about disease modification. And then finally, another group from the United States asking if this is something that we can reach. What's important is that Dr. Silver and Dr. Kalaji and Dr. Gislinger and others have already been showing us disease modification, guys. That's called interferon-based therapy in MPNs. And what you're talking about here is, can we harness the immune system, novel agents, older agents, in a safe way for you as the patient to add to what's already being done? And that's what I think what we can do, and we already have a precedent in our field. So as we look forward, the current phase 3 trials that are active and ongoing, so as I mentioned to you, we have to look at it in categories. So for patients who've already had a JAK inhibitor, you have the imetelstat. John nicely went through that for us this morning. That's the telomerase inhibitor. And that's an important one, John, as you pointed out. This is the only one in our field that uses the overall survival as the primary endpoint, suggesting that we're moving a little bit away from, and I think it's a good thing, to not just spleen and symptoms for you, but for your overall survival. Does it improve the length of your life? The momelotinib agent, as we talked about, that's the MOMENTUM study. Dr. V mentioned it yesterday, but just to remind you, that's the 4th JAK inhibitor that we're working on. The top-line data was shown to be positive, that it met all of its endpoints, but including anemia improvement. So this agent and the pacritinib agent, we believe, can not only be delivered in the setting of anemia and thrombocytopenia, but may actually improve some of those. And then the MDM2 inhibitor approach that John and Dr. Hoffman and their team have pioneered at Mount Sinai. We just heard about that. Then there's this category I mentioned to you, the add-on approach. This is new for many of you. You're already on your JAK inhibitor. Can I add in the second drug so you enter in the clinical trial, but you're allowed to stay on your JAK inhibitor? Well, that's nice. Then you don't have to worry about JAK inhibitor withdrawal or coming off a drug that you think may have some benefits still for you. And we mentioned some of those. The parsaclisib I mentioned to you, Navitoclax. I didn't mention the luspatercept. That's an important one to mention, which is to help the red blood cell count. So this can directly affect the anemia in myelofibrosis. In fact, this drug is already approved. It got approved for myelodysplastic syndrome, MDS, a particular subset for this exact indication, and we're actively testing it. Our friend and colleague, Dr. Aaron Gerds, is leading some of those efforts. So look out for that as an agent by itself or in combination with JAK inhibitor. And then, my goodness, as we talked about, the frontline combinations, that's what I highlighted here. So again, just to remind you, these pelabresib, navitoclax, parsaclisib, let the names roll off your tongue, right? John mentioned that a lot of these are just these are the generic names, much less the brand names. So that's one thing we need to improve on in our field. And then, and then also the pacritinib agent, which I really applaud. We have some of our pharmaceutical colleagues here. One thing I love about our field is that our MPN field is known for being one of the first to include you as the patients, patient-recorded outcomes. Here we are at the Mayo Clinic, where Dr. Mesa, Dr. Robin Scherber, Dr. Holly Geyer, Dr. Gowan, all of you that pioneered the symptom burden score, that's part of our clinical trials. Every FDA approval nicely includes that, and now other cancers are starting to do that. In addition to that, a lot of our pharmaceutical partners are engaged in longer-term follow-up studies to really help us understand the dosing, the safety, long-term side effects. So I really appreciate that. So even though you'll see FDA-approved drugs, some of the companies will extend studies in working with the regulatory agencies. So enroll on those if they're feasible for you and they work for you. So as I conclude here, I wanted to highlight this disease modification with the immune modulation approach. And Dr. Kalajian had nicely mentioned this Ruxo-PEG study, which was this beautiful study trying to take basically the two original drugs, right, the Rux and the interferon, and combining them. And he already showed us this data, which was an adaptive phase 1/2 study. But what I wanted to show you, in addition to the spleen reduction, is that they showed two important things. One is that they gave it safely. This concept that immune drugs or interferon is toxic to the patient, that hasn't been the case largely, at least in my clinic, Dr. Silver's clinic, and others, as long as you have monitoring, dosing, expertise, and education and dialogue. So they were able to combine the two drugs safely and feasibly, and you heard from him, there were no dose-limiting toxicities. But also the second thing is this, molecular responses. We're talking about clones, the molecular clones disappearing over time, and Dr. Kalajian was telling us that that can be durable over time, even in some of the patients who have treatment discontinuation. So again, part of disease modification, which Dr. Silver, you guys have already been showing for decades with this approach. And so it leads us for the setup, which is, can we utilize immune system modulation? Can we bring the immune system into these difficult to treat diseases like myelofibrosis? And at the ASH meeting, we were very excited to see this data. This is something that may be new for some of you. It's been mentioned briefly here at the meeting, which is the novel discovery of a new agent. This is a CALR mutated monoclonal antibody specific agent. It's got a funny name, right? Inca 033989. But what it is a selective sort of predator drone attack, if you'll allow, to the mutant CALR. So we're saying 20 to 30% of you, if you have ET or MF, have CALR, right? Dr. Nangalia, who presented day one, she's the one that discovered that exactly 10 years ago. Now, 10 years to the day, we have now in the lab and headed to clinical trials a specific mutant CALR monoclonal antibody. I want to show you this data because some of this is confusing, but I think the way that I can show it here may make sense. Basically, the problem is you have a ubiquitous target. Can you hit it without causing other issues? And that's what the team at Insight has done. It's a fully human, what we call IgG1 monoclonal antibody, so it looks like the other monoclonal antibodies that we're used to in cancer. It selectively binds to the mutant CALR, the problem issue, and it antagonizes this property, which then leads to all this abnormal signaling downstream to be reversed. Well, did it work in the lab? It sure did. They showed that it not only selectively inhibits cell proliferation and induces death of these bad cells, the mutant CALR positive cells, but also the investigators showed that they started to restore normal hematologic parameters and have molecular responses. Now, this is in the lab, this is mice models and ET, but what was powerful about this is not only hitting the target, but maybe the start of what? Disease modification. So the question will be for me, for Dr. Mascarenhas, we'll ask Dr. Silver as well, as he and I were talking in the break, if you can target CALR with all of these different pathways, so immune therapies, vaccines, CAR T, what's the best approach? This monoclonal antibody seems to be very promising. In conclusion, what I would say is let's remember that the JAK inhibitor combinations era, ladies and gentlemen, is here. I just told you there's 3 active global phase 3 studies. I want you to know that immune-directed therapies, what's old is new again. Let's not forget about interferon. Let's talk about the new ways of targeting these things. We need to optimize stem cell transplant, still the only curative modality. And then finally, I'll leave you with some of the active areas that we have all been discussing here, which is trying to focus on the difficult to treat areas of accelerated and blast phase post-MPN AML, not the subject of this meeting, but subject of our investigations, looking for targets and other pathways that may be active. And with that, I'd like to thank all of my team members at the MD Anderson, and particularly Ruben Mesa, whose mentorship, friendship, collegiality has not only brought all of us as investigators together, but all of you, all of us together as one MPN community and family. And I think without Ruben, we wouldn't all be here together. So with that, I'd like to thank you for your time and turn it back to John. Thank you.